Good morning. Good morning, everybody. All right, please come on in, find a seat, sit back and relax and enjoy the show. We're really excited for our 2023 version of our Acadia National Park Science Symposium. So thanks both to those folks who are here in person today and folks who are joining us virtually on Zoom. I'm Nick Fisichella with Schoodic Institute at Acadia National Park. We are a nonprofit science and education organization, and together with our National Park Service partners, we co-lead this research learning center. Our mission is to be a center for inspiring science, learning, and community for a changing world. And you all are really part of that work, and thanks for being part of science and our community of science here in Acadia. All right, I'm just going to do quick welcomes here. I'm supposed to warm up the audience, and then I'll be handing it over to Rebecca and Abe. All right, so who do we have in the audience with us today? So who's a scientist? Raise your hands. All right, and people on Zoom, you can raise your hands as well. And for those on Zoom who can't see the audience, that was what, 2/3 of us here in the room, perhaps. How about a manager? Who's a manager? All right, a smaller number, maybe 10% of who's in the room. How about educators? Who do we have for educators? Yeah, maybe a quarter of folks raised their hands, and on Zoom you can do the same. All right, how about volunteers? Anybody who's a volunteer? Yeah, I see a few hands going up. Awesome. And how about science communicators? Who are science communicators? All right, so for everybody on Zoom, I want you to know that every single person in the audience raised their hand when I asked who is a science communicator. Everybody raised their hands. Amazing. And for those of you in the room, just know everybody on Zoom also raised their hands. We are all science communicators. It's definitely something we all do in various ways. Some are professionals, and some of us do this, sort of on the side or personally with our families, with our friends. But it is definitely a role that all of us play, and I think this symposium is part of those efforts. All right, thanks for playing along with that. So the theme for today is restoration in Acadia National Park and beyond. So restoration, restoring, what are we restoring? I'm a forest ecologist, so everything is a tree to me. So if we're restoring Abies balsamea or Picea rubens, or maybe it's Liriodendron tulipifera, something fun like that to say. But this is really more, when we're talking about restorations, it's about more than plants or trees or wildlife or habitats. I think what we're really trying to restore is our relationships with nature. And this is one of the things I feel like I'm learning more and more about all the time, and perhaps is a major growth edge for me, that we're not just trying to advance conservation science and practice, but also engagement and even the language of conservation. Together, I'd say we are intentionally changing conservation science, practice, engagement, and language to be more inclusive, to be more positive, to be more forward-looking, and ultimately to be more effective. That's what we need to be doing. And conservation is a human practice, and so it must continue to evolve and develop. And change is hard. Let's not kid ourselves on that. And change's perpetual sidekick is uncertainty. And those are the two big things we're facing: change and uncertainty when we're thinking about conservation and cherished places like Acadia National Park. We know it's changing. We know the present already is different from the past, and we know the future is going to change even more. but when and where and how and which species. These are some of these big questions that we're, you all are trying to grapple with. And if Acadia can't remain the same as it was in the past, then what is successful conservation? That's a really challenging question in and of itself, and what I think the speakers will hear from today, and you all are grappling with that. And that truly is the leading edge of conservation. And I'm feel privileged to be a part of that here in Acadia. All right, so Acadia is one small place doing this really big work. And I want to thank you all again for being part of this community, and for inspiring science, learning, and community for a changing world. All right, so with that, I'm going to hand it over to Rebecca Cole-Will, who is the head of Natural and Cultural Resource Management with the National Park Service here in Acadia. And social science too, yes.
Thanks, everybody. Good morning. I'm just going to start with a good morning and welcome to you all from Acadia National Park. We always start with, it may sound banal, but this is Wabanaki homeland, this is Wabanaki territory, and we always want to acknowledge that at the beginning. But I think that over the course of the day, I hope you all get the sense of some of the vitality of that work that we're engaging in with Wabanaki colleagues. Kevin Schneider, superintendent, couldn't make it here today, and I know this is one of his favorite events of the year. He's, he would be having both hands raised as a science communicator, recognizing how important all of this work is for all of us. So again, he says hello and wishes he could be here. So I wanted to start with kind of framing how the Park Service and the federal agencies are thinking about this. And I thought about this a while, and then this came across my desk, and it would seem like it was prescient because just yesterday, the announcement about some of the new approaches that the Secretary of the Interior is promulgating for those of us in federal agencies. So new policies to strengthen climate adaptation and resilience efforts. And if you've been in a cave for the last 4 years, you might not know who this incredible person is, but of course that's Secretary Harland. And this photograph was taken when she was here 2 years ago as part of an incredible event that was hosted here at the Scooting Institute. And that beautiful basket that she is on her back that is fitting her perfectly was made by Gabe Frey, who is an incredible Passamaquoddy basket maker, cultural expert, who's going to be here this morning as part of some of our conversations. But that's birch bark and leather and ash, and it was just, I don't know how he figured it out, but it fits her, as you can see, to a T. So pretty incredible. So just as I said, just recently, the DOI is coming out with new policies, and a lot of this is what you're going to hear from us today around how are we in this climate emergency, in the climate crisis, what are federal agencies tasked with, and how we're thinking about how to manage in these really uncertain times. And so there are a variety that I just want to touch on quickly in terms of the thinking, and you're going to hear more about that, as I said, later today. So use science, indigenous knowledge, and landscape scale management. Adaptive management. What does that mean? A process of regularly evaluating the effects of management actions and resources. A lot of the work we're doing now is really adaptive in terms of thinking, well, we're going to try something. Well, is it working? Well, the ability to be more nimble in the way we think about things. And Abe's going to frame a lot of this more clearly with some examples as we move ahead. And then policy that emphasizes consideration of changing climate as the default for planning and decision making. That's pretty huge in terms of a sea change in the way we have been thinking as federal agencies around how to manage in real time. obviously, the need for high-quality climate information and climate uncertainty in resource management. So that's kind of the place where we also are living here. And the Schoodic Institute, as a science partner in terms of bringing that best available science as we move ahead, again, in a world where adaptive management and change is critical. And then, of course, landscape scale decision-making, and the notion of managed ecological transformation. So some of the work we're doing, and again, Abe's going to touch on this, is how we're thinking about making change, seeing what happens, and then moving ahead with with ecological transformation, recognizing that there are significant climate change risks. So, as I said, this is the leadership that's coming down to us, and I think that what you're going to see is how we're trying to implement that here at Acadia as kind of leading the way for some of this science. And then as part of that, I wanted to touch on some of the work we've been doing ourselves within our own planning and strategy. So just this year, we finished up a resource stewardship strategy here at Acadia National Park. This is kind of a standard process for national park units, and I want to give a recognition to the team led by Amanda Hardy at the Natural Resource Program that led us through some of this facilitated dialogue. This document, and it is a pretty substantial document that identifies sort of where we have seen ourselves in terms of identifying those issues, stresses, and threats, our priority resources, and what this has done for this in terms of thinking about the next 3 to 5 years. All my staff, raise your hands. This is kind of some of the working understanding that we have within the Division of Resource Management Acadia in terms of how we're going to work going forward. It's deeply integrated in terms of natural and cultural resources and social science. And it's also deeply interdisciplinary in the sense that nothing is, we're trying to get out of our stovepipes. That's not always easy. So something that's strictly natural resources or cultural resources, but ultimately all of our problems, our resources, and our issues revolve around how we think, work together, how we work with people and communities, and everyone who has a stake in this place that we call Acadia National Park. So I can't do more than one thing at once. I can wave my arms around, but I cannot move it. So what is a resource stewardship strategy? I'm not going to spend a lot of time on this, but as I said, it's a strategic plan. We look at this as really short term in terms of identifying whether those are issues, the stressors, and then what are we going to do about it in terms of goals and an integrated approach to kind of thinking through some of this strategic planning. So what we went through, and this is an example of just what are the priority resources and components. And like I said, you can look at this document. It's available on IRMA. And if you're like me and you have trouble sleeping at night, I can share one of these documents with you, which you could have for your very own. But take us through what are the resources and their components, so purpose and significance, mandates and research. And here's an example, and we're going to be talking a lot about these in terms of wetlands and salt marshes. What are the resources? What are the issues related to them? How do we think about those in an, as I said, a deeply integrated kind of way? For the first time here, this process was really important because it included cultural resource assessment. So there's something called a cultural resource stewardship assessment that led us through kind of, as I said, including cultural resource issues and the integration of cultural and natural resources. And we, throughout this, we involved, and I'm really proud to say that we had Wabanaki consultants The tribal historic preservation officers were on all of the meetings. And so we really heard a lot around, well, you know, what are the issues that are priorities here related to, you know, tribal concerns and co-stewardship, and starting to develop that. As I said, it's the stewardship goals and activities were integrated across all resources towards sort of the goals for stewardship. And the point here is the integrated approach to this and how we solve problems and how we're thinking. And that's where I'm going to turn it over to Abe to take us away.
All right, so I'm going to switch over and emphasize some of like how are we putting this into practice. So I'm Abe Miller-Rushing. I'm the science coordinator for the park. And so how are we actually applying that resource stewardship strategy? And also, I do want to encourage you, the resource stewardship strategy is online. You should go through it. It is substantial, but you can, you know, it'll give you an opportunity to find kind of where your research fits and where we're thinking. And you can both like see how you research might complement some of our priorities and some of the things we're thinking about, but also we welcome you to tell us what we missed. Like, feel free, like a big benefit of having all this research here is for folks to tell us like, I think you're wrong, I think you're missing this critical component. And so feel free to do that. But here I'm gonna talk about kind of how some principles that we're following as we're trying to put things in action on the ground. And I will say that we're not perfect at these. We're trying to make these the routine ways that we do things, and we're getting there. So we're on our way, but we're trying to get towards these things. So incorporating indigenous and Western science alongside each other, working with local communities. This is a picture of Rebecca with a local marine worm harvester, Fred Johnson. Yes, Johnson is his last name. And then taking a proactive learning-based approach, which we'll go through more later on, and then also adapting this, applying this resist, accept, direct framework to climate change adaptation. And just so you also see, here are some big projects that we have coming up. Like, these are going to happen. Some of them are already happening. And again, your research can happen alongside these. And so if you find some of the resource management staff during this, you can find out more about these projects. You'll hear more about some of the climate smart ecosystem restoration, invasive insect response like hemlock woolly adelgid we'll be working on over the next few years, rapid response to that, continuing bat ecology and recovery as much as they're recovering from white nose syndrome. Hopefully they'll recover from white nose syndrome. And then avian influenza, restoring aquatic organism passage, and so on. And so these are some of the things that are definitely happening, and I encourage you to kind of complement your work to it as much as you can. And I just wanted to mention too, so just how this is happening on the ground, that it's not all stovepiped. So those projects can all sound discrete and not really working together, but they are to some degree, and we're working to make these things work together. And one place where that is really highlighted is in Great Meadow. And so I'm just going to go through one quick thing just to show you how we're trying to put things together. And this is a restoration project that we're working on. Is collaborative between the park, Schoodic Institute, and Friends of Acadia, and supported through the Wild Acadia Initiative too. But there are lots of problems. So there's too small of a culvert, which is causing problems, causing a lot of flooding, blocking wildlife passage as well. There is ditching in the wetland as well, which is causing it to drain too much. So we've got both too much water at times, too little water at other times, and that kind of helped create a lot of disturbance-tolerant plants in there. You may be hearing more about this later on, so I'm not going to go into too much detail. But we also had invasive plants issues. We also have a big hemlock grove in this area, which is vulnerable to the hemlock woolly adelgid. And so we're taking this kind of learning-based approach to solve and trying to solve these issues and communicating. So following these principles that I mentioned. So we're trying to replace the culvert to fix some of the hydrology problems. Hopefully that'll also address some of the wildlife passage issues. We're doing removing some of the invasive plants and restoring native plants, taking this resist, accept, direct approach that you'll hear about in more detail later. And we're communicating a lot to the public, to visitors, as well as to, the town of Bar Harbor, where a lot of the water for those of you who don't know, Great Meadow flows out through Cromwell Brook right near the YMCA and right through Bar Harbor. And so we have to work with them as well on that. And then we're also working on cultural resources with the historic trails and vistas and the visitors that are visiting those sites. And so understanding how visitors are interacting with the restorations. So these kind of all things are coming together. And then we're also just starting a project to start to restore indigenous relationships with this wetland. And Suzanne Greenlaw will be leading a lot of that work. And with that, I'm going to hand it over to Rebecca to talk about how we're kind of taking this co-stewardship approach more broadly.
Okay. Well, we're almost out of time, but yeah, I wanted to point out that we're not there yet, but we have a vision and a plan to be more integrated in terms of creating a co-stewardship vision. So some of the projects that are going on, as Abe mentioned, Suzanne and the harvesters who've been involved with the Sweetgrass Project will inform on and help us build an agenda for understanding rematriation within some of these landscapes that we have here. I've been lucky enough to work closely with Bonnie Newsom, who's a associate professor at the University of Maine, around scaling out some indigenous archaeology and re-narrating the story of, from time immemorial, people have been here, but what does that really mean when you try to implement that on federal lands? And creating opportunities for better interpretation and education around that work here. And finally, we're lucky enough to host the Wabanaki Youth and Science Cruise yearly, and we're hopeful that some of that work will continue and expand with other opportunities for Wabanaki youth. So you've heard a lot about that work. In an hour when you come back after the poster sessions, we're going to have a longer session around some of the work that Suzanne and Dr. Michelle Baumfleck have done here. And then you're going to be the world premiere of a fabulous film that if you didn't know why this work is important, you will by the end of that, and then opportunities for some Q&A. So with that, if anybody has one or two or three burning questions for Abe. No? Okay, we will see you back here in an hour.
Yes. Yeah, so I just want to say, yes, so be back in here at 10:30. We'll start the next session right then. And any speakers in the audience that haven't loaded their talks for later on today, please do. And totally, yes, this is an amazing audience. You guys don't know how amazing you are, but go out there and meet each other and talk to each other. They're real projects, collaborative projects that come out of these meetings. They really do happen. And you're all very cool people. So enjoy the hour. So there's networking and posters, and then be back in here at 10:30, and look forward to getting to know you guys, everybody better. Thanks. Bye. All right. Hopefully everybody had a very good lunch and very good poster sessions. So welcome back. Thanks everybody for coming back. Really appreciate it. And so to round out today, we have a last session that's kind of a different aspect of how we're thinking about climate change, climate adaptation, and climate smart restoration. So the next series of 3 presentations will be really more Western science focused and looking at different aspects of salt marsh and wet and freshwater marsh and restoration, starting here with some of our invasive species management. And so talking about Nicole Collars, who's actually one of our second century stewardship fellows, now is at the University of Minnesota. Will be talking to us about a project that she's been working on for the past couple of years about glossy buckthorn genetics. And I'm going to hand it off over to you. Thank you very much.
Thank you, Abe. And it is such an honor to be here today with you all. Flew all the way from Minnesota and just really excited to spend a few days in New England. And I'm excited to share with you some preliminary results from work I did as a second century steward. And I'm going to start by acknowledging my colleagues who collaborated with me on this project, including Jesse Wheeler here at Acadia National Park, Eric Saka from the College of Charleston, who provided genomic expertise, and my postdoctoral advisor at Northeastern University, Dr. Randall Hughes. I also want to start by saying that the word invasive can be problematic language. Invasive and its synonyms can help reinforce nativist biases in human society. And as a scientific community, we haven't come to a consensus of what a better word might be. And so I'm going to use the word invasive, but I'm going to explicitly define it as an organism introduced into an ecosystem, either indirectly or directly by humans, that has harm, either to the environment or to human society. And we can see why the presence of invasive species in our national parks contrasts to the mission of the National Park Service, which is to preserve unimpaired the natural ecosystems and cultural resources that we have here in the United States. And consequently, the National Park Service spends millions of dollars and countless hours of labor. We have Emma here and Jesse here who are leading that labor here at Acadia National Park to try to eradicate invasive species in this park. And one of those invasive species is glossy buckthorn, which is a freshwater, it's really abundant in freshwater marshes. It's native to Europe and Asia, and it was brought to the US as part of the ornamental nursery trade. And we suspect, we don't have evidence for this, but we suspect that Mount Desert Island Nurseries, which was founded over by where Jackson Labs is today by George Dorr in the early 20th century, may be the source of the invasion on Mount Desert Island. And it's important to note that glossy buckthorn is reproductively prolific. It produces lots and lots and lots of seeds that have really, really high germination rates. So here in Acadia National Park, Jesse and his team have been managing both areas in the northeastern part of the park and down in Bass Harbor. And what I'm showing you here is each one of these blue shaded areas represents areas where they have treated glossy buckthorn. And this is data up to 2021, both in the northeastern part of the park and then also in Bass Harbor. And the way that they treat it is they will cut down stumps and they'll also spray the plants with herbicide. But despite their heroic efforts, glossy buckthorn reinvades areas that were previously treated or cleared. So what I'm showing you here is some, text that got a little out of whack, but a picture of Bass Harbor Marsh, and all of this is woody debris of buckthorn that was previously cut down and put into slash piles. And what you can see down here, these are all little baby buckthorns that are regrowing underneath those slash piles. And we were just out there yesterday afternoon, and it is a dense carpet of baby buckthorn at these sites. So for my second century stewardship project, I wanted to develop a population genetics data set to see if that information can help inform invasive management strategies. So before I dive into my approach and my results, I want to walk through how population genetics could help with invasive species management. And what I'm going to put here on this table on the left-hand side is a data analysis that I did. And on the right-hand side is I'm going to interpret how that data analysis can help with management strategies. So the first thing I looked at was the degree of genetic isolation among populations across the park. And this is going to tell me which populations look alike and which populations look different from a genetic standpoint. And that's going to be an indication of the amount of mixing that happens in the park. So if they all look alike, if there's really low isolation but high mixing, you know that they're probably moving around. There's probably a really high dispersal that's causing these populations to mix. And if that's the case, if buckthorn is present anywhere in the park, you're going to want to eradicate it because the chances are it's going to move from one location to the other. Another data analysis I looked at was I wanted to use the genetics to see if I can identify the source or sources of the reinvasion. So I can look at the genetic signature of the plants that I sampled from and compare it to the genetic signature of other populations on the park and see if they're a good match. And then finally, I wanted to measure the genetic diversity of each one of the populations I sampled. So there's decades of research out there that shows that the genetic diversity of a population can enhance its ecological performance. And we really don't want to enhance the ecological performance of invasive species. So if we can identify which populations might have especially high genetic diversity, we could prioritize eradicating those populations first over eradicating others. So the way that I approached this project was that I collected just over 300 leaves across 15 different locations within the park. I took those leaves back to the lab where I extracted the DNA from the leaves. And then I prepared those DNA extractions for a sequencing technique, which really what this sequencing technique does is it just takes the genome and cuts it up into a lot of little chunks. And then we take those chunks and we put them back together and we look for locations across the genome where individuals differ by a single nucleotide. And you might remember from your biology classes that DNA, like the code, the genetic code is made up of 4 letters, A, G, C, T. I had a t-shirt in undergrad that said AGCT spells everything because it is the basis of our genetic code. And so what I'm looking for when we assembled this genome was to look for different places across the genome where that A might turn into a G in some individuals. Or in some samples it was a T, but in other samples it was a C. And these are called single nucleotide polymorphisms, or SNPs. And so from this data set, we find, I think we identified 14,000 SNPs across the genome. So these are areas where there's variation among individuals. And after we filtered, we had to go through a filtering process where we get rid of samples where there's a lot of missing data, or samples where there wasn't independent, the different locations on the genotype or the genome weren't independent of one another. And once we did that, we had a data set of over 280 samples with over 7,000 SNPs. So these are 7,000 areas of the genome that showed variation across individuals and across populations. And so this is a map of the different sampling locations that I collected buckthorn from. So I collected from a couple different locations down in Bass Harbor, and then also the northeastern portion of the island. And each one of these populations is color-coded by just a subjective classification of region. And so we have the Bass Harbor here in red, the south part of the park in purple, and the north part of the park in blue, and then Jackson Labs in green. And the first question I asked was, what sampling scale explains the most amount of variation in my data? And this is going to help give me a sense of how much genetic mixing is occurring across the island. So what I'm showing you here, I'll go ahead and walk through this. This is the results of what's called an AMOVA. It's an analysis of molecular variation. It's very similar to an ANOVA statistical analysis in that it's taking these 3 different scales, looking at the among region scale, or among populations within region, or among samples within populations, and asking which level of that hierarchy of that sampling scale explains the most amount of variation. So what we found is that nearly 87% of the variation, this is just percent of variation explained, was explained by oh my goodness, I was giving you the take home already, among samples within populations. And so that tells me that there's more variation among individual samples, individual plants within a population than there is among populations or among regions. And so how I interpret this is that there's really weak genetic differentiation among regions and even weaker genetic differentiation among populations within regions. So there's lots of mixing. And this was actually pretty surprising to me. And when I first started this, I was like, oh yeah, I bet there would be lots of mixing. And I went and put seed traps out in the field because I'm like, okay, if there's lots of mixing, then these buckthorn seeds must just be raining from the sky. And so I put out these seed traps that my dad designed. He did a pretty good job. And they were out in the field from August through November. So when the fruits are starting to drop their seeds and towards the end of the season. But I only recovered buckthorn in 9 out of the 49 traps that I had put out. And most of the cases where I recovered buckthorn seeds, it was when there was a really intact canopy of adult trees. And like at Jackson Labs, where there isn't a lot of management. And so I had this impression that the seeds were just falling from the trees, directly from the tree to the ground. You know the saying, the apple doesn't fall too far from the tree. That's what I was thinking was happening with this buckthorn. So I got really confused when I saw how much mixing was coming up with this data. So I'm going to dive into the data a little bit deeper. And what I'm showing you here is a discriminant analysis of principal components. So what this analysis is doing is it's taking all that variation across all 7,000 locations in that genome and across all those individuals, and it's trying to condense it into two-dimensional space. And so what it does is I told it, like, okay, show me what the data would look like if there were 5 genetic clusters across Mount Desert Island. And each one of these is an individual sample. They're color-coded according to region, and samples that are grouped together look more similar than samples that are not grouped together. And so what we can see here, even though there's weak differentiation among regions, according to the AMOVA analysis, that Bass Harbor looks pretty different than the rest of the island, and that there is a little bit of differentiation occurring along this axis between some of Bass Harbor populations, but also between the south part of the main island and the rest of the island. So I want to dive in a little bit deeper and show you what this variation looks like in geographic space. So on the text again. On the left here is Bass Harbor Marsh, a picture of Bass Harbor Marsh, and each one of these dots represents an individual plant that I sampled. And then over here is Great Meadow. So we have Jackson Labs here, and this is Jackson Labs property that I sampled from. Great Meadow, where the culvert restoration's happening. This is at the end of Kibo Street, and this is off the boardwalk on Jessup Path. So what I want you to take from here is that this northeast part, this Great Meadow part, they all look the same. That green and that purple color, these are just representing different genetic clusters. They're all mixed together across those populations. But here in Bass Harbor, we have this like vomit color green versus this red. And there does seem to be distinct separation spatially. There's a little bit of mixing, but not nearly as much mixing as here. And if you haven't been to Bass Harbor Marsh, you can throw a tennis ball across this creek. And so there's more genetic isolation across that creek that you can throw a tennis ball across than in Great Meadow. Uh-oh, I got too clicker happy and now it's not advancing. Okay, let's go back to the old-fashioned way. Oh no, we're connecting to Zoom. We lost our connection to Zoom. Now we're frozen. I might have to like act out the rest of my presentation, get some volunteers to come demonstrate what my graphs look like. I think Chris might be that volunteer. Well, I'm going to keep going and then great, it worked. Okay. So this might look like a piece of abstract art to you, like it's really bold and colorful. So we're going to just spend a few minutes or 30 seconds, however long it takes for us to walk through it, just to orient you to what's going on in this figure. So this is showing the genetic signatures of each individual plant that I collected a leaf from. And it's telling us the degree of admixture, which is just that each one of these colors here represents a different genetic ancestry, like a signature, an ancestral genetic signature. And each one of the columns is an individual. And so you'll see that in these columns, an individual will have multiple colors, like in a stacked bar graph. And that tells me that, okay, for this individual, the probability, there's a higher probability that it came from this dark blue color than this yellow color. So it's giving us a sense of how these different individuals might be related genetically. And so each one of these individual columns is represents an individual's genetic signature. They're grouped together by population and then also grouped together by region. And so what I want you to get away from this is that Bass Harbor populations, they look different than the rest. And that's what we saw in that previous figure that grouped them to different clusters. And then what I also want to show you is, for example, Jessup Path here, that boardwalk population is pretty close visually to Jackson Labs. So that may be an indication that Jackson Labs is a source population for already at 5 minutes. Do I get a discount for the Zoom interruption? Do I get a, okay, awesome. Okay, oops, I jumped too fast again. Uh-oh. Okay, well then we're going to talk about this figure now. So this is a measure of genetic diversity at the population level. And what I want you to take away here is that, yes, it looks like some of these populations may be a little bit different from one another, but I didn't show you the error bars here. And these error bars are huge. And so really, this goes back to that initial AMOVA table. There's so much variations among individuals in a population that it swamps out any differences and genetic diversity across populations. All right, so Sarah will kindly advance the slides when they're ready to advance, but I want to go ahead and bring this back to my initial framework of telling you all how population genetics can help inform management. So what we know is that there's a lot of mixing, especially in the northeastern part. And that high degree of mixing, is it going to advance? Nope. That high degree of mixing means that if buckthorn's anywhere in the park, they're going to want to eradicate it. And as far as identifying the source of the reinvasion, there's so much mixing in these populations that that's going to make it a little bit harder. Yeah, thank you. And so because there's so much mixing, that's going to complicate inference, and I can't quite give you an answer of what the source of this reinvasion is. But as I dig deeper into the data, I'm going to look at the role of management history and the role of age class or height class of the buckthorn that I sampled from to see if I can clear up some of that variation. Next slide, please, Sarah. And then finally, there's not much difference in genetic diversity among these populations, so I wouldn't advise targeting any particular population out of fear of its ecological performance. Oh, we're not advancing. And so my take-home message for our team here at Acadia National Park is just to keep on keeping on, that they're doing a lot of great work and to keep on trying to get rid of buckthorn wherever they see it. And as I dive deeper into the analysis, if I gain any further insights, I will let Jesse and his team know. But for now, that's the message. But I do want to encourage you all to study Bass Harbor more carefully. If we can figure out what might be restricting dispersal across that creek, we might be able to mimic that strategy to help reduce dispersal at other areas of the park. And I have a whole big list of people to thank, including people in this room, such as Emma and Chris and Jesse, for all their great help. And I'm happy to take any questions, though I think I'm out of time, but maybe Abe would let me have a question or two, given the technical difficulties. Yeah, happy to. Oh, yeah. That's a great question. So Jean asked, who eats the berries of the buckthorn? And that is a really great question. At first, we thought it was birds, that birds were the big dispersal agents. But because I wasn't getting a lot of seeds in my traps, I'm like, they're not raining from the sky. So it's not like birds are chowing down on this and then just pooping lines of seeds across Great Meadow Marsh. It may be that small mammals are dispersing it. Then there also may be some abiotic agents of dispersal. So we're really interested in thinking about how these different locations are connected through waterways and hydrologically, to see if some of the way the water moves is moving seeds around. And some of this genetic mixing may actually happen from pollen dispersal. And that could be occurring through insects or through wind. So there's a lot more to learn here. Yeah. Yeah, that's a great question. So the question was, are there any visual differences in the genetics? So if can I look at the plant and tell that it has this genetic signature versus that genetic signature? And I don't have a good answer for you there, but I know that I've done a lot of work in seagrasses and salt marshes, which have really high genetic diversity despite just being one species. And you can't tell the difference among different genotypes just by looking at it. It's very cryptic. So I imagine buckthorn might be the same. Yeah. Yeah, that's a really great question. And I am going to pivot that one to our invasive species manager. He's like, no, this is not my talk. But the question was, are there any species or diseases that can be introduced to help?
We don't know of any, really. What we've witnessed in Acadia, there's a rust kind of a fungus that we've seen that doesn't seem to harm its performance at all. And so, yeah, on the subject of biocontrol, that's something that we look at, but there's not much that's readily available right now.
Thank you, Jesse. All right, any other questions, or is it time to switch gears? Okay. Yeah. Mm-hmm. Yeah, so that's a really great question. So the question was, what about time? How does time influence genetic divergence? And that could be a key player here. And when I dive deeper into these data, I am going to try to get at that a bit. And one other possibility is the reason that Bass Harbor is so different from the northeast part of the island is some of that could be because there were multiple sources of the invasion, that they came from a couple different nurseries, or maybe someone in Bass Harbor planted buckthorn in their backyard and it took off. So there's a lot more questions that we need to ask with these data before I give Jesse the final answer. Okay, well, thank you very much. I appreciate it.
You did awesome with that. Really, thank you. One thing I forgot to mention is that Nicole is one of our second century stewardship fellows. We've renamed the program and it's changed slightly. It's the Acadia Science Fellows. And actually it's open for applications right now. So if you know of early career from PhD student to early faculty that are interested, or if you are one of those people, I encourage you to look at the Skudik Institute website and apply. Has built many great relationships and great projects with us going forward, or so already. We've been really, really pleased with how that program's gone. And I'm looking in the back of the room. Hey, Sarah, or is it okay if we go ahead? Okay. I think the visuals aren't quite exactly right on Zoom, but we'll march ahead. So the next, all right, we actually are good. Okay, so next up, we have Tatia Bauer, who's going to be talking with us about salt marsh restoration that's been going on with the Maine Coast Heritage Trust and these SMART teams. I think you'll maybe tell us what a SMART team is. More than just smart. Anyhow, we look forward to hearing about your work. Thank you.
Thank you so much. Yeah, and this is going to be a really interesting complement, I think, to our morning session of Indigenous use and relationship with salt marshes, and now talking about the impacts of European settlers. So yeah, that's my little intro, and I am Tatia Bauer. I work with Maine Coast Heritage Trust as a regional stewardship manager, and I'll be talking about our salt marsh work. And so, that's fine. Luckily, okay, cool. So I don't think I'm going to go into too much detail about the importance of salt marshes. I think we can all recognize all their ecological values and ecological services they provide, and their cultural significance, as we learned again this morning. But I do want to highlight their carbon storage potential. Research is showing that that peat layer of salt marshes can store up to 10 times as much carbon as the same acreage of forest in Maine. So when we think of natural climate solutions to carbon sequestration, salt marshes really stand out. And so here's that SMART team that Abe talked about. So our partners in this work, really our guiding experts in this work, have been the Salt Marsh Adaptation and Resiliency Team, or SMART team, which is a kind of informal coalition of land managers, researchers, restoration practitioners within the Northeast that came together to decode and interpret the impacts of saute agriculture, which I'll be going into a little bit later. So they saw these impacts of saute agriculture on the way that marshes are functioning and developed restoration techniques to mitigate those impacts on the marshes. And I want to highlight specifically 3 folks that we've been working with the most, which is their design review team, Susan Adamovich, who was going to talk about this stuff, and I hope you all do hear this coming from her mouth because they're way deeper into it than I am, I will admit. But I will try my best. Also Jeff Wilson of Northeast Wetland Restoration, he's been our contractor, and then David Burdick out of the University of New Hampshire. And so saute agriculture, what am I talking about? Well, European settlers arrived, they saw these productive, vast, open lands that they could manipulate for their needs as they settled. And they called that, reclaiming marshlands. So basically, they wanted to block out the tides and salt water via these embankments, and you can see those guys working to build up that elevation so tidal waters wouldn't come in. They also systematically drained the surface with ditching networks, and so you can see that ditch going underneath the embankment. There'd usually be some type of valve that wouldn't allow the tide in, but would allow water to drain out. And those would both kind of combine to increase the productivity of saute on the marsh that was used, and other plants as well, that was used to feed livestock during that time, and really became this resource as settlements were created. And this is a quote from Susan, that all marshes even ones that you think are pristine, have been modified by European settlers at some point. So I just want to highlight that. That is something that she really wanted to drive home. And so all these impairments, all these modifications, I also wanted to say, like, those ditching would be an entire grid going up onto the marsh platform. You'll see a picture of those mapped out in a second. But all of those infrastructure pieces are now unmaintained. That practice kind of winnowed away in the 1900s or so. But they're still on the marsh, they're still altering hydrology, and we're seeing the impacts of that altered hydrology on salt marshes. So this is one picture. There's a really cool historical pictures out there. Essex County Historical Collections has a really good page about these practices, so I recommend going and looking at that. But very productive, these sites after the modifications. Nice. So I'm just going to go through some pictures of remnant modifications that we're still seeing out on the marsh. We got this plank drain with an embankment over top. We have, this is an old wharf site where basically the salt hay would be harvested. You could load it up on a boat and ship it off to whoever needed it. That's another embankment with ditching on either side. So the embankments were built up using the materials of the ditches. Which could range from 7 feet deep, this is a really extreme one, to really small, almost unnoticeable marginal ditches. And oftentimes, after they stop being maintained, they would kind of grow over. So you won't see these straight open water lines where there were ditches all the time. Sometimes the vegetation covers that up. But the underlying hydrological modifications are still there. So this is Old Pond, Marsh that's owned by MCHT and is undergoing a restoration project right now. And so this is just a close-up of that property. And so you can see some embankments and ditching, right? That big straight line across that was really changing hydrology. And so what are the problems with these ditches? Well, in these ditches, we have basically the ditch bottom is where the groundwater is going to go to. So it's lowering the groundwater table, which aerates that entire section of peat above the groundwater. And when that peat is exposed to air, oxidation, decomposition, and marsh subsidence. So these features cause the regions around the ditches to slowly sink over time, which is not great when you think of sea level rise. On top of that, those ditches often clog over time. They're not being maintained. Those clogs lead to pooling on the marsh surface, and that's another big problem that we're seeing. So I'm sure if you all have been out of a salt marsh recently, you've seen these pools. They're everywhere. It doesn't come out super clear in this picture, so I apologize for that, but they can look like this. We have pockets of standing water, pockets of vegetation, which will expand into massive mega pools. And so that trajectory of those like kind of pockets of pools becoming these mega pools is what we're really concerned about. And it's worth noting this is how it looks like regardless of the tide. So these pools never drain. They're just completely standing water at all times. And so that stagnation, the lack of flushing from the tides leads to vegetation die-off of all those plants that are really adapted to tidal cycles. When the vegetation dies, all the root matter within like the top 30 centimeters or so dies. So there's this rapid collapse of marsh elevation. And then there's also an element of anaerobic decomposition in those pools that cause further subsidence over time. And so we have these effects causing marsh subsidence. We also have sea levels rising. So eventually those are going to intersect. And right, that's going to be a big shift in how our current salt marshes are working. And so we want to restore marshes basically and revise that marsh subsidence trajectory so that marshes can build an elevation to keep up with sea level rise, so we have those systems moving forward. And so how do we do that? Well, first we go and figure out, we get into the brains of those farmers and map the features as we see them on the landscape. And so this is that old pond, that same site that I've been showing. And these are the mapped ditches, or these are the mapped embankments. And so you can see they're just everywhere. I mean, they're pervasive, not, yeah, you can't miss them. And so you can see they're also layered through time. So the orange was early period, you know, I cut off the time, but 1600 through 1700s, that was like that kind of orange color. That was their way of doing things. Then the next generation came in, they changed how they were altering the marsh. They went for the red. And then you have generations on top of generations. So it's really a complex layout. And you can see how much the hydrology would be modified by these systems. And so when we think about restoration, we want to rewire how water is moving off the marsh. And we do so kind of in two ways. The first is ditch remediation. So in areas where there is a high density of open ditching, we want to reduce that density. So we mow the salt hay, kind of place it into the ditch bottoms, which act as a substrate to capture sediment, raise the ditch bottom, raise the groundwater table to prevent that oxidative subsidence and decomposition. And you have to repeat that, you know, every 2 to 3 years, or you have to repeat that for 2 to 3 years, excuse me, to make sure that we just continually build up that elevation of the ditch bottom to halt that oxidative subsidence. And so here's a picture of that in the work. So we have a treated and an untreated ditch. And you can see that by year 3, the treated ditch already has, you know, alternate flora growing in it. It's not this open ditch bottom, which is really exciting to see. So that's one piece. We also want to address those pools, and we do so by channel remediation, which is where we dig runnels. In this situation, we're digging by hand, but you can also use equipment in the winter when the marsh is more solid, and using, you know, big mats to disperse the weight of the machinery. But we dig these runnels regardless, that are shallow and wide, so 30 centimeters wide, 8 inches, 30 inches wide, 8 inches deep. So they're shallow and wide, and we use those to basically reconnect those pools to the tidal flow. That tidal flow allows vegetation to reestablish, which again raises that elevation that's been lost, provides habitat for, you know, Nelson sparrows and other animals and wildlife. And those are kind of the two methods, with the ultimate goal of restoration to halt subsidence. So all those subsidence trajectories we want to stop. We want those pools to revegetate, which allows the marsh to recapture sediment with each tidal cycle, build up in elevation, keep up with sea level rise, and increase the resiliency of coastal systems to sea level rise, and also continue to store carbon, and more carbon, hopefully, and provide all the ecological and cultural benefits that we appreciate. So thank you for everyone who made today possible, and if you have any questions, I would be happy to answer them. Oh, so on time. That is an idealized graph, so the slopes aren't necessarily reality, nobody be worried. It depends on the system. So some systems have undergone a lot of subsidence, and so they're way closer to that point than others. And so the idea of restoration at this point is to revise it wherever they might be. And so there's kind of a statewide, we're trying to prioritize marsh systems to undergo restoration to kind of address ones that have the most potential to be restored to their full kind of function. But the time window is not that long, I will say, before things like thin layer deposition need to occur, which are a much more expensive and intensive operation. Oh, and I will say too, so naturally these systems, those pools will kind of break through over time and undergo this like secondary succession where what we're looking for in the restoration kind of happens naturally, but it just won't happen at the rate and in all the areas that we want it to. So we want to accelerate the process of succession basically in those pools. Anybody else? Yeah, absolutely. I omitted a picture, but yeah, there's, you can definitely, we have like timber mats that you basically lay out underneath the equipment. And for example, at the Old Pond site though, we did have to, last winter, do the hand digging of the runnels to stabilize the site because there's not much room between the pools and the upland. And so there's only so much space for the equipment to go. And we wanted to make sure it was really stable before we go out this winter to do the rest of the digging. Yeah.
Anything that has surprised you, for example, those disturbed areas that you're restoring, are you seeing a different suite of plants or anything that you didn't expect that was going to happen as a result of these restorations?
So we're in the middle of MCHG's first restoration project. A lot of this has been building on the efforts of the SMART team, so I can't quite answer that. But I will say that in that pool that we did hand digging on, already there are some plants coming in along the edge just in the first season, you know, Salicornia and that kind of thing. And you can see alterniflora wanting to creep in. So I think it just happened, that was way faster than even I kind of anticipated, yeah.
Have you been actively planting in the pools after you put in the ground?
No, and I think preferably we don't do that. You know, we're hoping that just the encroachment from the edges will kind of go into the middle of the pools. I think they have explored that in the past, just basically like spreading out seed heads of alterniflora and patens and stuff like that, and seeing what sticks. Yeah. Chris. Yeah, that's a good question. So if everything kind of goes to plan, it involves very little maintenance, you know, some, but you're not looking at like a yearly touching every single runnel. But it really is that kind of dovetailing of how water's flowing on and off the marsh, the width, the angle, maybe if you have like a grade, you know, everything kind of has to be pretty fine tuned, and that's a process. And so, yeah, it'll be interesting to see how that plays out over time, yeah.
Thank you very much. That was great. Thank you. So as Tatia mentioned, one of our speakers couldn't be here today, but we'll invite her up for another time. And so there will be another time to see her speak. So we're out to our last talk of the day, which is Jesse Wheeler and Chris Nadeau, who will be talking about our learning-based approach, so stuff we talked about earlier today, but our learning-based approach to restoration to different projects in the park. I'll hand it over to you guys.
Thanks so much, Abe, and thanks everybody for, clicker's gone, that's all right. Thanks everybody for sticking around for the last presentation. You made it, great job. And hopefully, like Abe said, I think we'll touch on a lot of things that kind of Abe started off with, right? So you'll kind of, hopefully it'll be a nice wrap up to the day as we talk about this kind of learning-based approach to restoration. And so I'm Chris Nadeau. I'm a climate adaptation scientist here at Schoodic Institute, and I work very closely with Jesse Wheeler, who's the vegetation program manager, hopefully I got that right, at Acadia National Park. And yeah, we'll be jointly giving this talk. And so I think many of you probably know that we're already embarking on multiple large-scale habitat restoration projects here in the park. And so, for example, we're restoring vegetation and soil on Acadia's mountain summits that have been severely degraded from decades and decades of human trampling and heavy rain events. And so, for example, how many folks participated in the Save Our Summits event? Yeah, look at that, awesome. So thank you all for participating in that. For those of you that don't know, the Save Our Summits event was an event where we asked volunteers to carry soil to the summits of Sargent and Penobscot Mountain for this restoration effort. And between that day that you all participated in and other efforts that we were able to have volunteers carry soil, we carried 3,750 pounds, over 3,750 pounds of soil to the summits of Sargent and Penobscot Mountains. And we'll be using that soil to start that restoration project very soon, this week, tomorrow. Other restoration projects that, have already been mentioned today, we're trying to restore, the Park Service is trying to restore natural hydrology to Great Meadow, and that's by replacing this undersized culvert at the outlet of the meadow, and also filling ditches, similar to what we just heard about in salt marshes, and purging old road beds that are changing the way that water flows through the wetland. And so we've been doing monitoring to kind of prepare for that. And then we'll touch on this a little more, and we've heard a little bit about invasive species already this afternoon, but we're starting to restore native plants in places where we have been removing invasive species. And Jesse will tell you a little bit more about that in a minute. But I think, like all restoration efforts around the world, we face a lot of uncertainties with these restorations that really threaten the long-term success of the restoration efforts. And so Firstly, we're applying a lot of novel techniques. And so, for example, our restoration on mountain summits here in the park, there hasn't been a lot of restoration on mountain summits in the Northeast, some, but not a lot, right? And so we're really having to learn what are the best practices for restoring soil and vegetation to these mountain summits. And then, of course, we haven't heard a ton about this today, although I think we've seen this exact picture, maybe last night, maybe it was today, I don't remember. But climate change is, climates are rapidly changing. We know that now. And that threatens the long-term success of our restorations. And there has been a lot of proposals for how we should be adapting restorations to make sure that they are able to persist under climate change. But really, almost none of those proposals have been tested. And so a recent review paper, for example, said that there's 145 different proposed strategies for how natural resource managers, like the managers at Acadia, can be adapting their management plans to climate change, but only 84% of those have no empirical support. And so they sound like great ideas, and in fact, lots of those ideas are being implemented out on a landscape all over the world, but really we have no idea if they work. And so, we need some information to know if these things are working. And really these uncertainties that we're facing, these novel techniques and climate change, in a lot of places, this is stifling action. So people don't want to waste money doing something that they don't know is going to work, or it's just going to fail in a couple decades because of climate change. And so here at Acadia, we're trying to take a different approach, and that's a learning-based approach to restoration. And what that looks like is that rather than just implementing one restoration strategy and hoping that works, we implement multiple restoration strategies at a time. We monitor those over time, we evaluate, and then hopefully we find the best strategy, or at least we have a smaller suite and new things to test in the future. But what this does, it allows us to move on with restoration that needs to happen without delaying, and learn, and not delay that restoration until we know exactly what we're supposed to do, which we will probably never know exactly what we're supposed to do. So this is kind of the approach we're taking here at the park, and I'll pass it over to Jesse to give you an example, and then I'll tell you a little bit more about some of the secondary benefits of taking this approach.
Thanks, Chris. So kind of give you an example here, a bit of a case study. I do want to set us to this nice scenery and just let's appreciate how the forests and wetlands at Acadia are exceptional. And it's fantastic that we have that, as our backdrop in our foreground, in our landscape. We also know that environmental stressors, including invasive species, pose a significant risk and harm to, in particular, our forests and freshwater wetlands at the park. And because of this danger, especially if we left invasive species and invasive plants unchecked to kind of repopulate the entire landscape, that the harm that they have displacing native plants and altering that landscape condition is something that we're not going to stand for as a park. And so to keep these landscapes resilient, meaning that they can kind of keep up with the natural change, we must control invasive species. So luckily, Nicole gave you a very good introduction to another character here, glossy buckthorn, so I won't go into too much into what this plant is. But again, like kind of what it's playing out, at least on the Mount Desert Island portion of the park, is that we find it in our wetlands, invading our forest understories as well. And so what that might be is displacing tree seedlings and kind of choking out our native wetlands. It's definitely, this particular species is our most aggressive that we find in the park. And we literally find it from the summit of Cadillac Mountain to the shores of our salt marshes and almost anywhere in between that it can get a hold of. And thanks to Nicole's work, we're getting a little bit more understanding of how seed really can spread. I guess it's hard to find the seed in your seed traps, but where it gets established, it will grow. And we have to stay on top of that. And so it's our biggest essentially management challenge that we face with invasive species control. We started efforts to remove glossy buckthorn in 2008 from the Great Meadow area, kind of near south of Bar Harbor. Shortly thereafter, Friends of Acadia recognized the important work they were doing and really supported us annually and continues to do so with that. Last year alone, our management for glossy buckthorn accounted for over half of all of our invasive plant management team's work hours. So we spend a significant amount of time on this particular species. And it also accounted for about 3/4 of the total invasive plant area that we managed out of more than 24 invasive plant species. So it's really a lot of our main focus, and you can kind of see here on the map, the little pink highlights. That's just an example from 2020, all the different work sites where glossy buckthorn specifically was treated. And then you can kind of see here where, we've been finding it in watersheds from where Great Meadow is located, the Cromwell Brook watershed, to Otter Creek, Cannon Brook, Kent Field, Duck Brook, areas of the North Ridge of Cadillac Mountain, and as of 2020 in Bass Harbor Marsh. Our staff spend a lot of time and resources managing and controlling glossy buckthorn with cutting, with herbicides, with manual control, and you can see here in sort of the image series here, just like Nicole presented, we come in, we get rid of the mature stems, we do a little bit of the kind of ground cover work. Yay, it looks great. Look at all this sunlight, it's fantastic. And then we're left with more buckthorn coming in. So after more than a decade of managing the glossy buckthorn in Great Meadow specifically, we've seen reinvasion every year. Recent control efforts at Bass Harbor Marsh in the last couple of years, after we've taken out kind of the canopy buckthorn and increased the light on the understory, we just find that with that full seed bank, a lot more seedlings coming up in that new light. So kind of where we're at now is where I'm feeling is something that's got to give, and it's either going to be us and our backs give out, or buckthorn. And like, so where where do we go next? Like, do we continue forth in the same strategies and methods, or can we implement something else? So this is where we're bringing in kind of the experimental adaptation approach of learning. And we figured, why not do this in our largest freshwater wetland complex, Great Meadow, and our largest salt marsh and wetland landscape at Bass Harbor Marsh. So I was thinking great scales and in areas where we're investing some of this work. And what this means, and this is part of this Wild Acadia landscape scale restoration initiative, what that means, luckily for me, is that we're not doing this alone. We have our partners at Friends of Acadia and Schoodic Institute to help us out with this. So that brings us to kind of what questions do we need to answer now, or kind of what things do we need to shift and think about into the future. The way I'm thinking about it is kind of like, what management tools should we be employing that are appropriate now and into the future, especially in a warming climate. So one example might be that we will not be able to rely on one control method. Herbicides, for example, are not effective when you're in droughts. And when we have so many droughty summers, it's really not going to be a tool that we can lean on as heavily, perhaps. The growing season's longer, which typically favors invasive plant growth over our natives. Our field teams are spread thin. We do the best we can to manage this, but we're just, as you saw that example of how much is across the landscape, we can only spend so much time in each place doing these kind of active, removal efforts. So what we're getting to now is essentially what can we do to slow down that reinvasion, especially what kinds of things can we think about for active restoration, and this would be behind buckthorn removal. And so this is where Chris and I and others on the restoration team, we're continuing asking for new help and advisors on this, is develop some new approaches to answer these questions. And so right now, what we have is finding these new tools to deal with glossy buckthorn. We've come up with 2 possible management strategies that we're going to test, that we're testing out after, as reinvasion of buckthorn is happening in these particular landscapes that I just mentioned. The first strategy involves planting, so basically removal, we're going to be continuing to remove these plants. And then what we're going to test is planting native graminoids, so grasses and rushes, where we're going to be planting those where we think that they will be able to spread quickly, on the ground and compete with glossy buckthorn for resources. The second strategy is to bring in shrubs and plant shrubs after removal. And we think with that, it can definitely, hopefully compete with glossy buckthorn for resources and light. And so help to kind of shade out, have that type of competition. And the way we're setting this up is we have currently 15 restoration plots within Great Meadow and Bass Harbor Marsh with this following design, where an example plot is a 10 meter by 10 meter where we fenced off so we can limit at least some deer browse pressure on the baby new happy little plants that we're putting up there to prevent deer browse. So that's where we're kind of setting up. That's what a plot looks like. Then we came in and we removed the buckthorn, kind of do an initial treatment. Some of these areas are in areas where we've been doing management for quite a while, and some for less period of time. And some are in wetter areas and some in drier. Anyhow, this is what it looks like. We use the normal methods we usually do: herbicides, manual removal. And then we delineated 4 subplots within there where we randomly assigned kind of where the position of each of those 4 subplots for the different treatments. The first one with the plantain graminoids, we have 9 different species that we used within that. The second treatment where we have shrubs, that's 9 different species of native shrubs that we planted there. And then we also have this disturbance only, or essentially an area where we dug holes kind of and pretended to plant things in there, but really just wanted to get at ground disturbance and see how much that might be playing into buckthorn kind of reinvasion. So we didn't actually plant anything in there, but we did a disturbance only. And then we have a removal only, or essentially kind of a control, sort of like what we are doing now as the 4th treatment. So we installed these just this summer, and we plan to expand the experiment to more plots next spring.
Awesome, thanks, Jesse. Yeah, so hopefully from that you can kind of see how, we're doing on the ground restoration, we're removing invasives, we're planting native plants, but we're doing it in a scientifically rigorous way so that we can learn a lot about what is most effective going forward. And I will just highlight that, Jesse and I get to stand up here and talk about this cool experiment and talk about digging holes, but the real people that dug the holes and are sitting in the back row conveniently in the middle there. So thank you to them for doing all that work. And so I'll just point out really briefly a few other examples. Yeah, it's perfect, thank you. I'll point out a few other examples about how we're using this learning-based approach. And this one follows up a little bit from something that was presented in a poster this morning. And the idea here is that we've taken Penobscot and Sargent, and we've broken them up into different zones. So you can see these green and white checkerboard here. And we'll be restoring in half of those zones and then leaving the other half for the zones alone. And that's really to try to understand what is the effect of restoration. And so we're taking restoration materials, we're putting down soil, we're putting down seed, we're putting down erosion control fabric, and that can have effects on things like small mammals. And so, for example, the poster today showed that small mammals are munching on some of our erosion fabric, right? And if that has a big effect on small mammals and then a big effect on the ecosystem, then we need to change what we're doing. And so this kind of taking this approach will help us kind of learn a little bit about how our restoration is affecting small mammals. And the other thing we're interested in this kind of approach is kind of visitor use management. So this is kind of a larger scale application of this kind of learning while doing approach. And then this is a smaller scale version. And so this is a plot on top of Cadillac Mountain. This is a restoration plot where we restored native plants and soil, but we're testing climate change adaptation strategies. And we're comparing that to non-climate adapted controls, or essentially what we're doing now. And so we're comparing 2 different climate change adaptation strategies. And I won't get into the details of that, but a few people have expressed interest in this over the course of the day from Sarah's great poster in the poster session. And we do have an example of this garden here at Scudic Point, right outside of Wright Hall. And so at the end of the symposium, if anybody's interested, some folks said they were, I'm happy to take folks over there and show them and explain this experiment. And it's a good way to get outside and get some fresh air and see some cool science. So obviously the benefit of these experimental approaches is learning, right? Reducing those uncertainties about what we should be doing, but there's also a lot of other secondary benefits to taking this approach. And the first one is what I call resilience through the portfolio effect. And And so let's say took a typical restoration approach, right? And we just thought shrubs is the way to go. We should be, in that Great Meadow restoration, that example that Jesse just described, we should just restore shrubs. We think that's the best way. And then let's say that we get a lot of heavy rain events, like we are these days under climate change, and the wetland gets too wet for shrubs. Well, then the whole entire restoration fails, right? And we've wasted all that money on restoring native shrubs. But if we take multiple approaches, right, like we are, and we plant shrubs and grasses, and then the wetland gets too wet for shrubs, then the grasses or the graminoids might survive that wet. And so we see some failure, but at least half of our restoration effort still persists under this uncertainty. And so, by implementing multiple approaches, we're hedging our bets against uncertainty and climate change. And in fact, we saw this play out in some of our plots that flooded in Great Meadow this year. And then the second big advantage of this, I think, is science and management communication. And so just to give you an example, a few years back, I had the opportunity to go with Park Service staff and others, Friends of Acadia staff, and help tour a US congressional delegation around the park, talk about climate change adaptation. And we visited many beautiful, all the most beautiful parts of the park that day with that congressional delegation. And Representative Mike Quigley decided to tweet to his 52,000 followers on Twitter, or whatever it's called these days, about our climate change adaptation experiment on the summit of Cadillac Mountain. And that's an amazing opportunity, right? And I think there's just something about the fact that we have these experiments, that people relate to these kind of experiments in a natural setting, in a national park, and it provides this really compelling opportunity for us to tell the story about how science can help us answer these management questions. And so I'll just wrap up by just kind of summarizing that, these long-term restoration success really depends on us learning and resolving uncertainties. And what we're trying to suggest here is that implementing multiple methods can facilitate that learning without delaying action, and it has these secondary benefits that just make it an even better approach. And then I'll close by just acknowledging, as Jesse alluded to too, that all of the work that we're doing is really a partnership between Scudic Institute, National Park Service, and Friends of Acadia, right? And that's an amazing partnership that we have that allows us to get all of this work done. And we have lots of other partners too. We're working really closely with lots of other researchers, and we consult with lots of other natural resource managers. And so we want to invite you that if you're sitting there in the crowd and you're thinking, oh man, I wish they would study this, and if you have the capacity or you are interested in studying that, then come talk to us about it. We're interested in partnering with other researchers and other natural resource managers to make sure that we're asking all the right questions and getting the most and learning the most out of these efforts. And I'll stop there and take questions if I have time. Thanks. Yeah, so the question is, what benefit, correct me if I'm wrong, what benefit is there of bringing in plants from other regions or other states and planting them on the summit of the mountains? Is that what you're talking about? Yeah, and I didn't talk much about that, but this is the project that if you're interested, come see it. There's an experiment out by Wright Hall, and we're trying to test this idea of whether we should be increasing genetic diversity, which is one of the most commonly proposed strategies for making populations more resilient to climate change, or if we should be getting warm adapted individuals or plants from warmer locations and bringing those to the summits and help the populations become more resilient to climate change. And that is the benefit, is that I think if we try to collect individuals from multiple locations, as opposed to what we do right now is we just collect seed from Cadillac, we plant it on Cadillac. And if Cadillac has low genetic diversity, then that could make it really vulnerable to climate change. And so if we go collect seed at other summits and bring it to Cadillac, then that could increase genetic diversity and make the populations more resilient. And I think more intuitively, if there's plants growing in warmer locations and we go get those and bring those to Cadillac, that could increase the resilience. Yeah, so far experiments aren't showing that, but we could talk more about that if you want to come over and see the plots.
Everybody want the last questions of the day?
Oh, man. I'll start. That's a great question, Angie. So, right, how do we think it, right? Because we're trying to take this a bit of more holistic approach in systems. As we're being more active in terms of bringing in plants, so we're still being careful and doing this in a way. I think that's where we're trying to build, as we build this, is equal to sort of what treatments are we going to do? What do we need to monitor over time, and what we need to kind of assess with that? So there's a lot of other things going on in these watersheds. It's been mentioned a couple times, particularly the Great Meadow area. So sort of that's a lot of a hydrologic restoration is in addition to the invasive species stuff. But right, what do we need to know kind of before a lot of that change has been made, the restoration that we need to know now, and track that over time. That's scary for me as a manager, right? I mean, like I don't want to be the 1600s farmer that like digs ditches and all the salt marshes and then thinking that was a great idea and then it ends up being horrible idea. So anything to add to that?
Yeah, I would just give an example, right? I think, as Jesse said, a lot of the things we're doing are small scale enough that, it's probably not gonna affect a bird population. But I think the summit example was an example of where we could be affecting small mammal populations, and at least we're trying to monitor that and restore in this kind of patchy framework so we're not affecting small mammals over the entire summit. But I think the Great Meadow restoration is another example of where, we are thinking really hard about, there are a lot of data from Kate Miller and others went into trying to know why that restoration has to be done. And now, yeah, we've had multiple meetings to try to figure out, okay, we're going to change the hydrology. What could that affect and how can we monitor that? So we're monitoring vegetation. There was a poster from Brooke today about monitoring birds with autonomous recording units. We're monitoring water levels. There's been some small mammal monitoring. We're thinking about monitoring beavers. So yeah, we are trying to track those things so that we're not exactly like Jesse said, so that at least if things aren't going the way we hoped, we know and we can make tweaks.
Thank you, guys. Thank you. So this last bit, so that was the end of the day. So I'll just kind of very fast wrap up. I just wanted to say like 3 things that I took home from this. One, there's a ton of amazing work. You guys are awesome. So thank you very much. That was my thing number 1 and the biggest take home by far. And then also I wanted to kind of point out 2 things that I think got highlighted by the 2 different sessions that we kind of had going on. One is that hopefully we were able to show you that we really are working towards integrating or doing indigenous science and Western science together. And I know that really gets us to think a lot about revisit how we do science and how we think about relationships and how we think about relationships with the landscape. And that'll be a long time for us to change how we do our work kind of going forward. And so that's one take home. And I think we'll be chewing on that for a very long time and adjusting how we work. And the other thing I think what we wanted to really drive home is this, what the subject of this last talk is, this learning-based approach, that we're really trying to learn as we go. And of course, that puts an obvious role for science. Like we're not going to be able to learn as we go without the science and without the work that you guys do. And so there's lots of opportunities for us as the science community for Acadia to kind of build on some of the management actions that are going and also identify some of the things, like I mentioned at the beginning, some of the things that we might be missing. So feel free to tell us when you think we're missing something, but also feel free to build on the things that we're doing and let us know, and so that we can kind of really maximize effort and maximize the learning that we're getting. And I guess the last thing is that we really want to communicate both, all of these kinds of take-homes from today. And as Nick was talking about at the beginning, everybody in here is a science communicator. We're also bringing in the next generation. I don't know if any of you saw on the way down or back to lunch, we have a new Schoodic Education Adventure group, which is a middle school group coming in for 3 days of science education and environmental ed here. So kind of getting them while they're young. And so I think that was all I wanted to take home from today. And then so last housekeeping kinds of things. So we'll be sending out an email with the attendee list. If anybody doesn't want their emails included, just let Emma know. But that way you'll have Emma Albee, who is doing a lot of the communications for this. But that'll be a way that we can stay in touch with each other. For posters, we'll be asking anybody who presented a poster if you're willing to share that, and then we will post it to our IRMA data store, so it could be archived as a part of the proceedings for this science symposium, so the future folks can, folks that couldn't attend, or the future folks can look back and understand a little bit more about what was going on. The recording, so today's proceedings were recorded. The recording should be up in a couple of weeks, and so you can look for it on the Skudik Institute YouTube page. And my last things are some thank yous. I want to give a round of applause for all of our speakers today. Thank you very much. And a big thank you to the Skudik Institute staff that did a lot of the logistics, all of the behind the scenes work and organizing for today. It actually takes a lot of work to make today go. So I really want to thank the Skudik Institute staff that really helped to do that. And lastly, I want to thank all of you for coming and for all of the work that you do, and for really helping us. It really is a vital part of informing the management to protect Acadia. Science is written right into the founding legislation for why the park was protected in the first place, and we're really continuing that now. And so thank you for helping us continue that tradition. And I look forward to working with you over the next weeks and months and years ahead. Thank you very much. Safe travels home.